T lymphocytes are central to the cellular adaptive immune response, but need to be activated and educated by dendritic cells or other antigen-presenting cells in order to achieve their effective functions. When we discussed the role of dendritic cells, we described the three signals that came from the antigen-presenting cell to activate the T cell. So in this video, we are going to look at the other side of that interaction and look at what happens to the T cell upon activation, as well as some more general T cell biology. So just to remind ourselves, the dendritic cell or other antigen-presenting cell passes three signals to the naive T cell. Signal 1 from the MHC peptide Tcr interaction, signal 2 from the co-stimulatory and co-inhibitory ligands and receptors, and signal 3 from the cytokines acting in a paracrine manner on their receptors on the surface of the T cell. And the nature of these signals is determined by the integrated environmental signals that drove the activation of the dendritic cell in the first place. So this is where we got to previously, with the activated dendritic cell having travelled down the afferent lymphatics to present the antigen to the naive T cell in the paracortical region of the lymph node. And just to remind you that the lymph node has a highly organised architecture, fed by multiple afferent or incoming lymph vessels and drained by the efferent lymph vessel. The B-cell germinal centres in the follicles are surrounded by the cortex, and then the paracortex surrounds the medulla. And it is in the paracortical region that the dendritic cells encounters the naive T lymphocytes. So let's have a look at what is happening on the other side of these three signals in the T cell. But to do that, we really need to understand how the T lymphocytes are produced and end up in the lymph node. T cells, like all hematopoietic cells, originate in the bone marrow, where they become a lymphoid precursor, and they have a commitment to becoming a T cell. They then migrate to the thymus, which is an organ that sits just above the heart, where they mature, and once they have undergone a series of checks, they are released from the thymus and migrate to the lymph nodes, with some ending up in the spleen and gut-associated lymphoid tissue or GALT. Now remember that although these T cells are described as mature, they are still naive, as they have not encountered their cognate antigen. These T cells will sit in the lymph nodes and wait until a dendritic cell with a complementary MHC peptide combination comes along and activates them. Taking a closer look at the thymus, we can see the highly structured architecture of this organ, with a fibrous capsule surrounding the cortex and the medulla, with the thymocytes, which are these T cell precursor cells, entering the cortex from the circulation, where they interact with cortical epithelial cells and start to differentiate into a mature T cell as they move through the thymus into the medulla. Looking more closely at this process of T lymphocyte development and maturation, we have a hematopoietic precursor which is showing early signs of commitment to the T cell lineage. When this lymphoid progenitor enters the thymic cortex, it is a double negative or DN cell, meaning that it does not express either CD4 or CD8, which we have defined as the surface markers that identify a helper or cytotoxic T cell respectively. We know that these cells are likely going to end up becoming T cells, but at this point we do not know whether they will end up being a CD4 positive or a CD8 positive T lymphocyte. T cell development is elegantly organised, both spatially and temporally in the thymus. And as pre-T cells move through the cortex through the double negative 1 to double negative 4 stages, they will start to proliferate due to the influence of a cytokine called interleukin 7, and they will start to rearrange the genetic material that encodes their T cell receptor, and will begin to express a unique T cell receptor on their surface. Once a functional TCR has been formed, they will start to express both CD4 and CD8. So they transition from being a double negative T cell to a double positive T cell, expressing CD4, CD8, and a T cell receptor. The double positive cells are screened against the cortical thymic epithelial cells, which express both MHC class I and MHC class II molecules on their surface, to see whether the TCR can recognise our own MHC molecules. If the double positive cell has a strong interaction with MHC class I, it will be driven towards becoming a CD8 T lymphocyte. If it has a stronger interaction with MHC class II, then it will commit to becoming a CD4 T cell. If it cannot recognise our MHC or does not receive sufficient co-stimulation, then the T cell will undergo apoptosis in a process we call death by neglect. So now we have a CD4 or CD8 committed double positive cell. But these double positive cells have a relatively short lifespan of only a few days and need to be rescued, which is the process of receiving the right stimulatory signals, or they will undergo apoptosis. These signals include engaging their TCR with self-MHC self-peptide complexes, and the purpose of this is to ensure that the T cells are able to interact correctly with MHC class 1 or MHC class 2, which we call positive selection. If they pass this step, then they are allowed to become a single positive T cell, expressing either CD4 or CD8, depending on which lineage they have previously committed to. However, in the next area of the thymus, the medulla, we need to ensure that the interaction between the TCR and the self-MHC self-peptide complex is not too strong, as this would indicate that the TCR is potentially autoreactive and recognises a peptide of auto-origin. So any cells that interact with self-antigens too strongly are screened out from the T-cell repertoire and removed by apoptosis to try and ensure that we do not go on to develop an autoimmune disease. And this process is called negative selection, or sometimes central tolerance. The cells that are responsible for this are bone marrow-derived antigen-presenting cells and medullary thymic epithelial cells. And only cells that pass both positive and negative selection are released into the peripheral circulation. And it's estimated that the maturation process takes between 2 and 3 weeks, with about 95% of the potential T cells being removed from the repertoire. But we still generate a staggeringly diverse population of naive CD4 positive and CD8 positive T cells. Just to summarise these processes, positive selection is checking to ensure that the TCR expressed on the CD8 or CD4 committed T cell can interact with self-MHC, expressing a small fragment of self-peptide. We do this to ensure that the T cells can recognise the MHC molecule. And this is mediated by the cortical epithelial cells that have all the cellular machinery to express both class 1 and class 2 MHC. If the cell does not receive the correct signals, it is not rescued and dies by neglect to be removed from the repertoire. If the T cell passes positive selection, then it undergoes further checks to ensure that it does not recognise self-peptides expressed in the MHC too strongly or too weakly. And cells that do not pass this check are removed by apoptosis. This process is driven by bone marrow-derived antigen-presenting cells and medullary thymic epithelial cells. The bone marrow-derived APCs have come from different areas of the body and present peptides from different tissues, while MTECs have a property we call promiscuous expression of tissue-resident antigens. This is under the control of 2 key genes called AIR and FESF2. So they're able to present peptides that are normally only found in very specific tissues, for example the liver, and any T cells that are potentially autoreactive are removed by this process of negative selection. The overall goal is that the T cells that are released into the circulation are both tolerant to self and restricted to self-MHC, so as not to potentially trigger autoimmunity. Now this process is not perfect, and individuals do unfortunately sometimes go on to develop autoimmune diseases, and there are a huge range of reasons for this. But in the context of a self-reactive TCR, the body needs to strike a balance between removing potentially self-reactive T cells, but also providing enough coverage to be able to defend us against pathogens. And so, on occasions, T cells that are self-reactive can evade this screening process, and then, in combination with other factors, can lead to the development of autoimmunity. The threshold for Tcr affinity is still one of the mysteries that immunologists are trying to figure out about the immune system, and so far we have not been able to quantify this threshold. It is in effect a bit like Goldilocks trying to find the right porridge. We don't want T-cell receptors that bind too weakly or too strongly, we want ones that have just the right affinity. But what this affinity is remains to be determined. Now, negative selection does a great job of removing autoreactive T cells, but as I've said, some do slip through the net. So the body also has other mechanisms to induce self-tolerance. A rapidly growing area of interest for immunologists is regulatory T-cells, or Tregs, which directly and indirectly suppress the proliferation of autoreactive T-cells. Tregs can develop in the thymus and appear to be an alternative fate for autoreactive T-cells, so instead of dying by apoptosis, they may commit to becoming a regulatory T-cell instead, but the precise mechanism underpinning this is as yet unknown. This population of Tregs is referred to as thymic Tregs, and another population can develop in the periphery with subtly different functions. But regulatory T-cells can be identified by the presence of the master transcription factor FOXP3, and they partially mediate their effects by the secretion of anti-inflammatory cytokines such as IL-10 and TGF-beta. As a further layer of protection against the activation of autoreactive T-cells, the body has evolved several other mechanisms of protection. Perhaps the simplest and one of the most effective is to hide the potential antigen, so that it is not presented in the context of MHC, and therefore the autoreactive T-cell cannot see it. If the T-cell does not receive the correct combination of signal 1, 2, and 3, it will not be activated. A final mechanism is peripheral energy, whereby a T-cell that receives signal 1 from the MHC peptide TCR interaction, but fails to receive a suitable co-stimulatory signal, will become unresponsive or anergic. This may be due to the absence of an appropriate ligand-receptor interaction, as shown in the left-hand diagram, or due to the presence of co-inhibitory molecules like PDL1 and PDL2, as shown in the right-hand image. So we have started to talk about the different subsets of T-cells in this video. And just to remind you that it is a combination of signal 1, 2, and 3 that is received from the activated antigen-presenting cells that allows the naive CD4 or CD8 T-cell to develop their effector functions. And although this diagram shows the CD4 cells, the process is the same in CD8s, just involving MHC class 1 and CD8 instead of MHC class 2 and CD4. The CD4 positive and CD8 positive T-cells that leave the thymus are mature but naive, as they have yet to encounter their cognate antigen. However, they are highly mobile cells, and it's estimated that each T-cell recirculates from the blood circulation system into the lymph circulation system and back every 12 to 24 hours, so they are constantly moving around the body searching for antigens. And this high degree of movement increases the likelihood of the T-cell encountering an antigen-presenting cell displaying a complementary peptide. If they do encounter a complementary APC in the secondary lymphoid organs, then they become activated and start to replicate, producing multiple copies of the original cell in a process termed clonal expansion under the influence of a cytokine called interleukin-2. Further cytokines then drive these clonally expanded cells to become effector cells, or long-lived memory cells, that provide the immunological memory. Effector CD4 positive T-cells secrete numerous cytokines that activate other immune cells, while CD8 effector T-cells are cytotoxic and lyse infected or cancerous cells. CD4 positive helper T-cells can be further divided into subfamilies depending on their master transcription factor and effector function. We have already encountered some of these in a previous video, but many more families than originally anticipated exist, with discrete effector functions. The differentiation is driven by the signals received from the antigen-presenting cell and the cytokines that polarize the response. If the naive T-cell is exposed to a combination of IL-12, interferon gamma, and IL-18, then it becomes a Th1 cell, which is responsible for cell-mediated immunity and inflammation. Th1 cells are mainly active against intracellular pathogens like viruses and some bacteria, and have been implicated in tissue inflammation and autoimmune conditions. Th2 cells result from polarization with interleukin-4 and interleukin-2, and are mainly concerned with antibody-mediated immunity. They are active against extracellular parasites like Helminth's worms and activate eosinophils as a result. Because of this, they have been implicated in allergy and asthma. Th17 cells arise from stimulation with IL-6, IL-23, and TGF-beta, and are characterized by the master transcription factor ROR gamma T. They secrete different forms of interleukin-17 and interleukin-23, and play a role in barrier immunity against extracellular pathogens such as bacteria and fungi. They are an inflammatory phenotype and have been implicated in autoimmunity and tissue inflammation. T-cells, as we discussed, dampen down the immune response and provide part of the peripheral tolerance, including lymphocyte homeostasis. Peripheral Tregs, not to be confused with those thymic Tregs we described previously, arise from stimulation with TGF-beta and interleukin-12 and mediate their downstream effects via TGF-beta, interleukin-35 and interleukin-10. This regulation of Tregs has been implicated in an impaired anti-tumour response. The last subset we are going to describe are the follicular T helper cells, and these, unlike the other subsets discussed so far, tend to remain within the lymphoid organs, where they are interacting with immature B lymphocytes in the follicles and germinal centres to drive B cell maturation and facilitate class switching, which we will talk about when we look at B cells in more detail. More recently, however, two further subtypes have been identified, Th9 and Th22 cells, and these both seem to have a role in defence against extracellular pathogens. Activated CD4 positive T helper cells play a fundamental role in the activation of cytotoxic CD8 T-cells and help tailor the effector functions towards an antiviral or anti-tumour response. So as we can see in this cartoon, the dendritic cell is simultaneously inducing the effector functions in CD8 positive and CD4 positive T-cells in the lymph node, and the CD4 positive T-cells are then providing additional signals to the CD8 positive cells to enhance their response through surface-bound receptors and the release of specific cytokines. Once the CD8 positive cytotoxic T cells are fully activated, they will leave the lymph nodes and migrate in the circulation to the site of viral infection or to the cancerous cells, and through cell-cell mediated contact release active mediators including perforin and granzymes onto the surface of the target cell, which induces pore formation and cell lysis. These helper CD4 cells enhance this response and help activate the cytotoxic cells and ensure that their response is appropriate. The last concept we are going to look at briefly in this video is the formation of T-cell memory. Memory cells are long-lived and allow us to respond more quickly if a pathogen is ever encountered again, which is the secondary immune response. Interestingly, memory T lymphocytes seem to have less stringent activation requirements compared to naive T cells. Naive T cells are almost exclusively activated by dendritic cells in the lymph nodes, but memory T cells can be activated by B cells, macrophages, or dendritic cells, and are much more sensitive to stimulation. They also have a very different distribution pattern in the body. Some remain in the lymph nodes, but others are circulating amongst the tissues or reside in specific organs, perhaps because that is where they originally encountered the initial threat, and so they are primed to defend that same entry point. There are distinct subclasses of memory cells, and 4 have been identified in humans to date: central memory, effector memory, resident memory, and stem cell memory. The differences between these are beyond the scope of this video, but they occupy different regions of the body and have different expressions of surface markers that allows their classification. The important thing to remember is that both CD4 positive and CD8 positive T cells can form memory populations, with CD8s being more abundant. But many questions still remain unanswered about these fascinating cells, such as how these populations are maintained over the duration of our lives and how they coordinate their efforts to mount an effective secondary immune response. That was a more in-depth look at the activation, expansion, and effective functions of T lymphocytes. These cells are central to the adaptive immune system and orchestrate the cellular response. Of course, these cells work in tandem with the B lymphocytes and the humoral response, and that is the topic of the next video.